had a PhD in Cambridge. And I said, yes, I would very much like to have a PhD paid for. What are embryonic stem cells? That sounds interesting. I have no clue what that is, but I can probably do that.
Stanley Strawbridge is a leading expert in stem cell biology. He did his PhD at Cambridge University. Now he is at the University of Sheffield, building his very own lab from the ground up. Today he's going to sit down with me to answer some of the field's biggest questions. So what is an embryonic stem cell?
Two things. So a stem cell is defined by two features. You have this idea of self-renewal, right? So that's the ability of a cell to give rise to two daughter cells where one of them has the same potency as the mother cell. Potency is the other component here. That is the ability to give rise to a more specialized cell that is more restricted than the mother cell, right?
How would it decide which one to turn into? Like, is the body sort of put out a request in a way? Like, how would it work?
So this is one of my favorite questions, and one I work on, and one that a lot of people work on, right? There's a couple different ways that I think about this.
When they do IVF and they have the embryonic cells, do they have to have that sort of activation signal for them to start? Or are they already on that starting process?
So IVF is literally taking an egg and mixing sperm into that. There's a couple different flavors of how you make embryos in the dish. One of them is injecting a sperm into an egg, and that's what drives the actual initial activation.
Looking at your research, let's say you find an answer about why these cells are changing the way they're doing, and even though it seems random right now, you find the cause. Why is that important to find the answer?
If we think about it from kind of the nerdy version, and the thing that got me excited in the first instance, there's some really fundamental things happening with this question. The one statistic that I find staggering is that at 30, I think it's 32 years old, if you have IVF treatment and you have an embryo successfully make it to the blastocyst stage, you have a 1 in 3 chance at a live birth. Over the next 10 years, those statistics drop down to less than 10% success rate.
So thank you, Stan, for joining me today. I wanted to get started by sort of looking at your academic journey and sort of how you first got into science.
This goes all the way back to high school for me. So my first time in a research lab was a summer studentship for 6 weeks at a local military base where I was doing bench chemistry. How old was I? I was 17 years old, and I was put into a lab with a mass spectrometer, right? And so this is a thing where you put a little bit of liquid in, and then it goes through this very, very long tube at a high temperature. It's like a little oven. And then as things go, different compounds will move at different speeds through it, and they'll separate out, right? So that's what chromatography is.
Okay.
And then eventually it'll go, so that's the chromatography part. And then at the end, it hits a detector and it'll be fragmented. And then it'll tell you what the spectrogram is of the mass. And somebody let me play with this for an entire summer. And I thought it was just the coolest thing. And I was hooked from the get-go. And I ended up working there for, 3 summers and then a year after undergrad. So this was once upon a time, before I was a stem cell and developmental biologist. I was on the path to becoming a scientist in kind of chemical and biological weapons defense. I know, it's a very logical career progression, right?
Yeah.
So Homeland Security actually paid for my part of my undergrad.
Really?
Yeah. So I got a scholarship through them, and I did a different summer job up at Plum Island Animal Disease Center, working on foot and mouth disease virus. And after my undergrad, I had to go work in support of the homeland for a year to satisfy the work requirement attached to my undergrad scholarship. Uh,
And how
and
did you feel working there? Did you decide then that that's not what you wanted to do?
so in the background, I was doing that, but also through undergrad. So I did my undergrad at West Virginia University in the U.S. And in the U.S., you can do 2 degrees, and you can do research throughout undergrad. So it's, I mean, two very big things that are different about the American and British education system, right?
Mm.
British education system, what you get 1 summer of research if you're lucky, and then one year of a project. You can't just show up and say, "Hi, I'm a first year. Yeah, can I please hang out in your lab and play pet some things?" Which is what I did.
Yeah.
And I was in labs all throughout my undergrad, and I was doing both biochemistry and mathematics as an undergrad. So I basically started out as a biochemistry student, and then I just kind of kept taking math classes. And I got to the point where I was just like, well, this is a math minor now. And then eventually I got to the end of my second year. I was like, I'm just going to keep taking math classes. So I'm just going to make this a math major now. And then I had both of these, and I wanted to figure out a way that I could combine my mathematics background with the experimental side. And I realized that what I was doing uh, in the bioanalytical lab didn't do that for me. And I needed a bit more, like, I needed to be able to do a bit more with the mathematics that I had.
Mm.
And that's what took me over to the UK in the first instance to do an applied mathematics degree. And I was like, well, I need to learn how to do mathematical modeling because I want to model biological systems. So I took a master's. All these UK degrees have fancy, names. So this one was math modeling and scientific computing.
Yeah.
And I was in Oxford for a year there, uh, trying to learn how to be a better mathematician. I can say without a doubt that I did become a better mathematician, but if I was a better mathematician, I would probably just be a mathematician.
Mm.
But unfortunately, the power of what I'm able to do is the combination of my skill sets.
Mm.
And that's kind of, I went from just a mathematician to an experimentalist who has a quite wide tool set, which includes, being able to program and make mathematical models, do quantitative things, high level of like statistical abilities,
Mm.
which is quite useful in modern biology, right? Because we've moved away from just these, let's go knock out a gene, to, okay, you've knocked out the gene. Maybe it's affected the digit tip length. I'm just saying something arbitrary.
Yeah.
How is it affected like the digit tip lengths? And like you're looking for statistical significance of like a phenotype on the order of, I don't know, something quite small.
Mm.
And that's what we're kind of, those are the kind of things that we're trying to do, is get those kind of quantitative bits of information. After that, I saw that Microsoft had a PhD in Cambridge, and I said yes, I would very much like to have a PhD paid for by Microsoft, doing some type of modeling with an experimental component. What are embryonic stem cells? That sounds interesting. I have no clue what that is, but I can probably do that.
So what is an embryonic stem cell?
So, two things. So, a stem cell is defined by uh, uh, two features. You have this idea of self-renewal, right? So that's the ability of a cell to give rise to two daughter cells where one of them has the same potency as the mother cell, right?
Mm.
What is potency? So potency is the other component here. That is the ability to give rise to a more specialized cell with that is more restricted than the mother cell, right? So I work in embryonic stem cells. This is what's called a pluripotent stem cell. Um, this can differentiate and give rise to all of the cells, including the germline. That means, you know, uh, sperm if you're XY, eggs if you're XX, sex chromosomes, and all of the tissues in the adult body, bone, blood, muscles, you know. Uh, so you have different types of potency. That's pluripotency. You can also have, you know, bipotency that'll give rise to two different can give rise to cell type A or cell type B. You have some cells where they can only self-renew and give rise to one other cell type. So there's all sorts of different flavors of stem cells, and they span from uh, things that are proper uh, stem cells, which will self-renew for the entire lifespan of the organism. And you'll find these in the adult. This is like what happens in homeostasis, right? So homeostasis is we need to make sure that your skin is skin for the entirety of your life.
Yeah.
And if you don't keep self-renewing and differentiating, your skin falls apart and you have a very bad time. Where there's other types of things that look like stem cells, but they're more transient in their renewal. And this is what we would call progenitor cells. So they self-renew for a short period of time to make sure you have a big enough population of the cells, and then they'll start to differentiate. And then the self-renewal capacity goes down and they differentiate.
Okay, so you have the stem cells, and it's almost like a cell that can turn into how would it decide which cell it will turn into? So they can turn into like a blood cell, like a sperm cell, or how would it decide which one to turn into? Like, is the body sort of put out a request in a way? Like, how would it work?
So this is one of my favorite questions, and one I work on, and one that a lot of people work on, right? So uh, there are a couple different ways this can happen. So you have something called a niche. There's a couple ways. One way is a niche. So you might have a cell that sits very close to a group of cells, and it might hang out there. And as long as it's in this neighborhood, it will keep self-renewing. But, and this could be from things like diffusible proteins, like things that are floating around in the intercellular space. It can be from literal physical contact and them transmitting, or, cell surface contact. And the minute they start to move away from that niche and stop losing that signal being produced over here, they will start to differentiate, right? So this can happen where this cell will divide and you'll give rise to a second cell over here, and this one will move away. This one will differentiate, but this one will keep self-renewing.
Would it naturally float away? Would that cell just naturally sort of, like, why would it move away from that group of cells it's already with?
So in the egg chamber, or the of the fly, where you make oocytes, this is literally just because you've had an asymmetric division away from the niche, and this cell is now far enough away, and then there's just kind of a continuous pull down there. I don't know if it's mechanical. It must be mechanical. I'm not quite sure about that, or if it's kind of some type of fluid flow, but I doubt that.
Yeah, maybe just when it splits apart, like the forces are just naturally sort of just keep going and pushing it away.
Yeah, I think I'm almost certain somebody's done work on this. I'm just not read on that. So in the early embryo, it's a little bit, which is where I work. So where we get embryonic stem cells from is you have a thing called a blastocyst, right? So you have an egg is fertilized by a sperm. That becomes a zygote. So we now have two copies of the genome, one from the mother, one from the father. And then the egg goes through cleavage divisions, which is literally just the cell dividing in half without getting bigger again. So you go 1 to 2 to 4 to 8. And then right around that 8-cell stage, it starts to kind of compact in, and, the outside becomes smooth. And that's what's going to go on to make your cells that become the placenta. And the inside cells then make a second fate decision to become either the cells that will make the fetus or the cells that will kind of support the fetus. And that's a second tissue called the hypoblast.
So, when you have the sperm cell and the egg, and they come together and fertilize, how does it mix? So is it the DNA which carries all the information, and how does if I'm correct here, how does the DNA mix into making this like final product of the?
So yeah, so this is a process. So you have the maternal genome in the egg, and you have the paternal genome in the head of the sperm, right? When that hits in, you have two nuclei at that point, effectively, and then these things have to merge, and this then becomes diploid. So when you have activation of that, then basically the entry of the sperm activates. I'm trying to think of a simple way to say this.
Yeah, that's
Uh,
okay.
yeah, you it basically starts a bit of a wind-up toy,
Mm.
where you kind of say, all right, you know, we've now had the sperm fused with the egg, and there's a whole bunch of biochemical reactions that start to happen as a consequence of that say, start to do your thing,
So would there
right?
be inside the DNA or the genome? Would there be like almost
Inside the
like
cell itself.
would it be a set of instructions that almost like the DNA would hold, and then once it combines, it sort of activates that cell of those instructions?
So in the cell, you have more than just DNA. So DNA is like an information carrier, but you need someone to execute. Uh, you know the you need something to execute the program of development, right?
Mm.
And so you have a couple things. So have you heard of the central dogma of biology?
No.
So it goes DNA to RNA to protein, right? So there are three big macromolecules that are are at work here. So DNA is that information,
Mm.
uh, and then proteins are the ones that are doing the work, right?
Mm.
So these are things like enzymes that catalyze reactions. They also uh, deal with how genes are expressed. Um, and that intermediate step, RNA, when you say a gene is expressed, what does that actually mean? What it means is you have a piece of DNA is what's called transcribed into RNA, right? So messenger RNA. You might have heard this from like the messenger RNA vaccines that everyone got
Mm.
during the pandemic. So these are bits of degradable nucleic acid that carry instructions into the cytoplasm to make proteins,
Okay.
right? And they're only there transiently, and so are the proteins. So it's not like a stable integration into the genome, right? So everyone who's getting a bit worried that these are going to these vaccines are going to do something weird to our bodies, um, sure, there are side effects, but you're not actually altering the genome with that kind of thing. Uh, so you have DNA, proteins then induce, they read the genome, they produce these RNA transcripts. These then go out, a thing called the ribosome runs along those, and then they produce a protein. And those proteins are then responding to the fusion of the sperm with the egg. So that starts up this whole process. And I'm talking whatever 20,000 genes there are, times however many copies of proteins there are. I mean, it's an astronomical number of molecules we're talking about within a single cell, right?
And how
And
long
they're
would it take to sort of do this process?
uh, so in mouse, um, cleavage is 2.5 days. So from one cell to 8 cells, that is
Mm.
2.5 days.
Okay.
Uh, and to make a blastocyst, where we get to, that is 4.5 days. In the human, that's 7 days. So there's slightly different time scales here.
Mm.
Uh, and so the to circle back, So you have these niche-driven cell-fate decisions, but then you also have non-niche-driven cell-fate decisions, right? So, for example, that compaction step where you make the cells that will make the placenta, that's positionally driven. So it's only the cells on the outside that are going to start to say, "Oh, I'm on the outside. I know I'm on the outside. I should probably change my cell fate." Cells on the inside, they change their cell fate. This is something that I'm trying to figure out, and the community is trying to figure out. It's a group of cells that exist. They're homogeneous, right? They all look the same. And then all of a sudden they start to change their cell identity randomly in what they call a salt and pepper pattern. Why are those cells changing randomly? We know that there is a chemical signal that's doing this, that's diffusing through, but there's no spatial pattern, right? There's no niche. There's no spatial information. They're not doing it in kind of like this side goes one way, this side goes the other way. What's happening there?
So there's an activation chemical that goes through the cells and then they start changing randomly into the one. Would it be something to do with how, this is just like complete, just off the top of my, just some guesses here, but could it be something to do with how sensitive each of the cells are to that chemical? Or you know how like in electronics you get different resistances of things. Could they be resistant to, some of them be more resistant to the chemical? Like, could you measure that sort of sensitivity or resistance?
So, couple hypotheses. Some of my favorite ones. So there are all the cells are producing the signal. All the cells have receptors to respond to the signal, right? Are you expressing different amounts of the receptor, right? This is your ability to respond to it. Why are you producing different amounts in a heterogeneous fashion? So then that gets back to the chicken and the egg situation. Why are you producing different amounts of the receptor in the first instance? Why is it the ability to actually transduce the signal even though you have the same amount? Is it a non-uniform field of chemical signal? If it is the case, then how did it get non-uniform in the first instance when it should be? So these are all sorts of things I'm trying to work towards. One of the methods, one of the simplest things to address at the moment is the field uniform or non-uniform? And the easiest way to do this is literally take a microscope or take your cells, label the protein. So it's 22 kilodalton protein, right? So not small but not big protein. We have a thing called antibodies. So these are things you produce. You can also use it for like immune therapies. So any drug ending in MAB, monoclonal antibody. These are things that kind of look like little, I don't know what you call them,
little prongs.
Yeah, little prongs.
Yeah.
And they'll go in and they'll specifically recognize things and they'll stick onto them. And what we can do with that is we can put dyes on the back end of those, and we can shine light on them. They'll emit light back out to us, and we can look at that under the microscope. Okay. So in principle, that's a great idea. There are 23 FGFs, 3 of which of those are expressed at the same time or quite near in this point. FGF4 is my favorite. But then you also have FGF3 coming from a different tissue, and then soon FGF5 will all come in. And one part of the protein all looks very similar, and it's only a small part of the protein that looks different. There isn't a good antibody to do this. I've tried that, doesn't work. That was time in my life. So then I said, let's try a different thing. So there's this cool technique called super resolution microscopy. And I found a friend who later became my boss. Um, and I said, this guy's name's Stringent Basu. He was in Cambridge. We were in Cambridge together for a while. And then he's down at Imperial now. And I said, Stringent, I want to image this thing. Can you teach me super resolution microscopy? Can we do this? And I got a fellowship through the Wellcome Trust for this project to try to label. So go into the genome, put a protein on the back end, right? So you take your bit of DNA,
Yes.
and then on the back end of the DNA, you can insert another chunk of DNA. And what that'll let you do is that will let you image it. So have you ever heard of fluorescent proteins?
No.
So there are things called I think the first one was green fluorescent protein. So some very clever people won the Nobel Prize because they said that was it algae? That algae glows when we shine light on it. Be really interesting if we could isolate that and express it. They did it, and then people were like, it'd be even cooler if we could put that in other organisms. Again, they did that, they expressed it, and then people started tacking that on to the back end. Literally the chunk of DNA into mammals and human cells, and you can get, now there's a whole zoo of fluorescent proteins.
But why would you want those fluorescent proteins on the back of a DNA?
Or on the protein. So it's the bit of DNA says, I now want the fluorescent protein. This bit of your genome now has the code to make the protein. So when you make that mRNA, it goes out, makes the protein. When it folds up, you have your protein, but then you also have the fluorescent protein. Now I know where that fluorescent protein goes in the cell. Is it in the nucleus? Is it at the cell surface? Is it in the cytosol? Is it in the endoplasmic reticulum? What part is it subcellular localized to? Is it in my cell of interest if I'm in an organism, right? Is it in the liver? Is it in the blood? For me, the question was, what part of the embryo is it going to, and how uniform is it?
So you can track where it goes because of this fluorescent part that will almost like, it will literally lights up in a way. So it'll go around the body and you can actually track, all right, this is, and why would it be important to track where it goes?
So this is getting to my question about what the field of the molecule looks like, right? Why is that cell changing cell identity, right? Is it because like something that's cell intrinsic, that is it some way that the cell is responding to the signal, or is it the actual signal itself and how the signal is distributed? And I wanted to do that, but it turns out these proteins are almost, twice as big as the actual protein I'm trying to label itself. So that's kind of messed up how well the protein can actually move around. And
I see.
It's an imperfect system, but it still gives you information. And with that, instead of just trying to image the protein itself, right? Because you can image the entire thing and everything will just glow. Or you can do a thing called this super resolution technique, and that's you image one protein or one fluorophore at a time over time. So you take lots and lots of snapshots, and it's called a couple different things, but the one that I think makes the most sense is a stochastic optical reconstruction microscopy. STORM, right? So we like acronyms in science. And one of the reasons why STORM is an apt name is because when you actually watch these videos, it literally looks like dots flicking on and flicking off over time. And so as a single image itself, there's actually not a whole lot of information there. But when you have, an hour of 10 millisecond images, and then you press them all together, you can then start to build up a picture of what's there.
So these are the cells that you were talking about before, where they like activate randomly, like the salt and pepper. So I'm just trying to put it in context for my understanding here. So that would be when you have the stem cells, like the embryonic stem cells, and they're specializing into different things, or what are they changing into there? Is that what you're talking
Yeah.
about?
So this is I've got. Is there an ability to like show you pictures?
Yeah, you can share your screen, I think. Yeah, yeah.
Let me see if I can do this. This might make a bit more sense. Got some lovely cartoons here.
Some nice animations.
I'm a picture-oriented guy.
Okay, yeah, I see that.
So this is kind of your 8-cell cleavage. This is where you have these cells. These blue ones are what's going to make your placenta. And these white ones called the inner cell mats. This
Mm.
is what's going to start to specify in this random pattern before it resolves into this, which is called the epiblast. So these are pluripotent cells, and this is where we get embryonic stem cells from. So you have the epiblast, which is kind of what's called in vivo. So that's in the organism. And then if we take those cells out of the embryo and put them in the dish in vitro, then those will grow up, and that's what's called embryonic stem cells.
Okay, so let me
Yeah.
clarify here. So we have the sperm and the egg, which is back before these photos, and then they're dividing up to the 8-cell one,
Mm-hmm.
and then after that you have they sort of separate, and you have the blue ones which create sort of the outside cell wall, placental wall almost, and then the inner white ones are they just reproducing, constant, still replicating.
Yeah. So this is a like an unspecified progenitor. So it's going to go on and make that second fate decision to make these two cell types.
Okay, and then the two cell types, those are the ones, those are what you call like the embryonic stem cells, the ones that are going to sort of be shoot randomly going into now different specialized cells.
Yeah. So this is where this is a tissue called the epiblast, and that is where embryonic stem cells come from. But if you were to let this keep going in development, then that's what's going to give rise to all the other tissues of the body.
Okay, okay, that makes sense. So you're looking at how, so sort of what triggers these, how they turn into different cells, and then why they do it at different, even though they will receive the activation signals at probably the same time, why they're changing at different times.
Exactly. Exactly.
Mm.
And then I think I have some nice pictures here. Yeah. So these are some nice embryos. So this is after they've formed up. So you can see this is a immunostained. That's those antibody labelings, and you can see each one of these is a nucleus, little fuzzy nucleus. So that's the epiblast cells and the primitive endoderm. And then down here we actually have the whole embryo labeled. So you have the blue is the trophoblast. Those are your placental cells, and then your other two cell types there. You'll
Inside,
notice
yeah,
that
go on.
these little heads, these little snowman-looking heads.
Mm.
Basically, there's a little proteoglycan capsule here called the zona pellucida. That's basically an eggshell. So all embryos, we all live inside of an eggshell, and then we have to hatch out. And so there's a little bit that gets weak, and then it starts to pop out, and then the embryo has to push through. And what you're seeing here is the embryo starting to push through that capsule.
Is that sort of the divide, or
Yeah, exactly. So this is literally like there's a little hole there that the embryo is literally squeezing through, and then yeah. So I
wow?
was like a grandpa, I like to say at the end of talk or at the beginning of talks when I'm explaining this, and just like chickens, we all have to hatch outside of our eggs to before we implant.
Wow,
Uh,
that's so interesting.
it very rarely do people laugh at that, but I say it anyway because it makes me laugh.
Yeah, and so when we're looking at each of these, like, so because this is really early on of life, right? You've got the sperm and the egg, and we're so right at the beginning. Um, when I think about our fully human body now, we have like the central brain, which is feels like it's making the decisions at least.
Hmm.
When we look at these early cells, is there one main part that's making decisions for the other cells, or are they all sort of like on an equal plane where they're all making their own decisions to sort of replicate and do the tasks that they're sort of doing and the instruction they're doing?
So this is kind of getting at this concept of what's called self-organization, right? So the embryo does this thing where it does the work itself, right? Um, there's kind of a program. So there's this one cool study where in sea urchins. So one of the early embryological studies in sea urchins was can't remember who this guy was. He basically got it at the 2 cell stage, and he took a hot needle and he stabbed one of the cells and killed
Mm.
it, and he left it like that, and he got half of a sea urchin. Right. I think the experiment goes if you remove it, then you get a regular sea urchin, and it's
Mm.
basically you lose information and there's no like auto regulation happening. So in mice, and I can say this for mice for certain because I don't think we have done this in humans. So those pictures I was showing you, those were mice. You can tell by the whiskers. Um, Old man joke. Yeah, I'll be here all week. Um, uh, if you take an embryo and split the morula, that 8 cell stage embryo, in half and let it grow up, it'll make a small but fully formed blastocyst, right?
Mm.
That's that final stage with the 3 tissues. You can then transfer that back to a pseudo pregnant mouse, and it'll give birth to live pups of a normal size.
Mm.
If you take two embryos, two 8 cell stage embryos, and put them together, you will get one mouse of a normal size, but the blastocyst will be very big.
Mm.
So there is some type of autoregulation. There are feedback mechanisms within the mammalian embryo where these cells are always talking to each other. It's small enough that information is moving across these cells in a manner, um, whether it's through direct contact, through short range signaling, or just kind of propagation of information
Mm.
from one cell to another, kind of indirectly, where one cell is like, I'm on the outside while I'm on the inside, these kind of things. So there's a few different ways that this is definitely happening. Um, they it's through this autoregulation, through diffusible signals, positional information, that the embryo is really able to do its work.
So when we talk about this sort of like communication between the cells, um, what would that look like? Would it be some sort of like because would it be some sort of electrical impulse that's sent, or would it be they give off a chemical and the other cells receive that chemical and interpret it in a certain way? Like, what would it look like?
Yeah, so most of it is either through. So there's a couple different. The main way that cells talk to each other is through receptors. So things that sit on the cell surface and are literally looking for things. Like when they come into contact with a thing that they're able to interact with, a signal is transduced across the cell membrane, and then a big cascade of information goes down and affects how that cell behaves, right?
Mm.
And receptors, cell surface receptors, can interact in multiple ways. So they can go through diffusible ligands, so small proteins that will go out and diffuse around. Um, there are cell surface receptors or there are ligands that are uh, in the cell surface that will, like, delta notch is a famous one. This one will cleave off, but it's stuck in the cell surface until it does that, and that's direct contact mediated. Um, there's also what's called kind of the extracellular matrix. So cells produce lots of proteins like collagens, laminins, these really long proteins that give structure, and cells can interact with that.
Mm.
Um, so
The
there's
receptors themselves, would they be a protein as well?
they are
Yeah.
indeed a protein.
Okay. Okay, that makes sense. So, looking at your research, let's say you find an answer about why these cells are changing the way they're doing, and even though it seems random right now, you find the cause. Why is that important to find the answer?
So um, there's a couple different ways that I think about this. One of the If we think about it from kind of uh, the nerdy version and the thing that got me excited in the first instance,
Mm.
um, there's some really fundamental things happening with this question. Cell-cell communication, right? What's called morphogen gradients, right? So a morphogen, this FGF signal, these diffusible ligands. Um, there's this guy called Alan Turing. Do you know this guy?
Yes.
So
Yeah. Yeah.
he put out three big papers in his life that were kind of pivotal. One was on the uh, what was it? The computing machine. What's that one
The
called?
Enigma machine, is that what you're talking about?
Yeah. So he put out one on like basically this is what a computer does and how you add numbers in a very sophisticated way, right?
Mm.
The next one was his artificial intelligence paper, where we get the highly popularized Turing test.
Yeah.
And the one that is incredibly important to every mathematical biologist, but not so much to the rest of the world, is a paper called On the Basis of Chemical Morphogenesis. And in this paper, he goes through and through simple uh, coupled um, partial differential equations, he's able to get to the point of saying why we have animals with spotted bodies, but not with spotted bodies and striped tails, but no animals with striped bodies and spotted tails.
Mm.
Can you think of an animal that does that? So a cheetah, spotted body, striped tail, not the other way around.
Mm.
You can have a zebra. I mean, there's animals with straight bodies and striped tails. Um, it comes down to domain size, right? So basically, if you're on a big domain, you'll get stripes. If you're on or you'll get spots, and if you're on a small domain, you'll get stripes.
When you
Um,
say domain, you mean like landscape, like area to live in? Like, what do you mean by
literally the body of the animal, right? So
that? Okay.
whenever the pattern of that animal is laid down, and then you let it grow, if you lay down the pattern on a big domain, then uh, uh, you get stripes.
Right.
So
Okay.
like the body of the zebra is bigger,
Yeah.
or sorry, the body of the cheetah is bigger, so you get spots. The tail of the cheetah is small, so you get rings.
Mm.
Um, and then I'm doing this a disservice. There's this mathematical biologist Philip Maney, who does a beautiful version of this, um, and he then poses a question, something like, okay, well then elephants should have all sorts of crazy zigzag stripes and neon fluorescence, and his point is that it's not just the domain size, but it's also when the pattern is laid down, right? So
Mm.
if you lay the pattern down when the organism is small, then you're not going to, yeah. So, um, so Turing's whole point in this paper, and there's a lot of work that is like, okay, well, maybe it's not a perfect system or a perfect model, but there is information within there, and it's basically, a thing is produced, it diffuses, it goes away, it's taken up, and you can basically have a model for how cells interact with each other that way. Another guy came along, Louis Wolpert. He came up with his French flag model, and it basically explains how if you have a signal that kind of looks like a little exponential, so you have a point source and you have an exponential distribution,
Mm.
you can actually have a system where you have three different cell types arise.
Mm.
And the reason for that is thresholding, right? It's not about, um, it's about the relative amount that of the signal you see, right? So you don't have continuous change, but you have discrete change in the cell identity. Um, so these are some really fundamental questions that we think about in developmental biology and why things become what they are.
Mm.
Uh, if we want to start thinking about um, like use to the world
Mm.
and like just beyond the purely academic, um, IVF is quite important, right? So assisted conception, in vitro fertilization, um, pregnancy. Why do embryos fail to develop? Um, is it something cell intrinsic? Are we failing to produce a certain cell type? Do the cells not generate the correct patterns? What is happening here that prevents them from doing the 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 right type of organization that needs to happen for them to proceed? Um,
Do they when they do IVF and they have the embryonic cells, do they have to have that sort of activation signal for them to start? Or are they already on that starting process?
so IVF is literally mixing sperm, taking an egg and mixing sperm into that.
Mm.
There's a couple different flavors of how you make embryos in the dish. One of them is just mixing it. One of them is injecting a sperm uh, into an egg, and that's what drives the actual like initial activation
Right.
of
Okay.
the process. Um, but the one statistic that I find staggering is that um, at 30, I think it's 32 years old, if you have IVF treatment uh, and you have an embryo successfully make it to the blastocyst stage,
Mm.
you have a 1 in 3 chance at a live birth.
Mm.
So 66% failure rate for an IVF patient at 32. As you get older, those over the next 10 years, those statistics drop down to less than 10% success rate.
Why does it drop so significantly?
I mean, the reasons for this span from, time of egg collection, like did you actually have uh, a good egg? Like, uh, what is the genotype of that egg? Is
Mm.
it euploid? Euploid being having the correct number of chromosomes um, to health of the mother. You know, not only are you able to generate a healthy embryo is one question. Is the mother in a healthy state to allow an embryo to implant and to
Mm.
develop, right? Um, so it's getting to that stage, but then the next step is let's assume we do have a healthy embryo develop. Um, implantation is the next and highest point of failure. And the best data that we have is from this guy Snow back in this, I think it was the 70s. I don't quote me on that one. I can't remember what year that was. Um, but it's something like 70% failure rate at the point of implantation. And these are what's called chemical pregnancies. So you're able to detect that an embryo did try to implant, but there's no clinical signs beyond a blood test,
Mm.
right? There's no site of implantation. So those two things together are what we're trying to get to understand, well, why is that such a high failure rate? Is it the health of the embryo? Is it the status of the mother? Is it the status of the embryo? Why what is the synergy between the fetal maternal interface that is preventing normal pregnancy from proceeding?
Yeah, that's so interesting because I'm just thinking about why, like, as a woman gets older, why that failure rate increases, and I'm imagining it as almost like when you get pregnant and you're growing a baby inside of you, it's almost like an energy leech to the system in a way. It's like if you obviously the body's trying to protect itself at all times, and if it thinks that if having a baby could cause a failure of the whole system, maybe it sends and activate a signal to that embryo to say, okay, don't keep the process going because the whole system can't survive this process perhaps. So maybe if you, when you identify what is like why this sort of activation is happening, or why the failure is happening, maybe you might find that it's the mother's body sending a signal to the embryos to say, don't keep this process going because it's going to damage the whole system.
Yeah, so I don't pretend to be a reproductive physiologist. I'm much more on the embryology side of things.
Mm.
Uh, but I definitely think it's just kind of a not so much that as it's more just a natural part of aging,
Mm.
uh, and um, kind of hormonal status and just kind of permissibility uh, of pregnancy. I don't know if it's so much. I mean, there are certain uh, there's they're called the great obstetric syndromes, right? So um, recurrent miscarriages, miscarriage syndrome, preeclampsia, um, these kind of things, where it's kind of like, well, why is that happening? A lot of work is going into that, um, and I think there are a lot of ideas out there surrounding this.
Mm.
Uh, and I think there's still quite, but basically, women's health is wildly understudied historically, right? Um, and it's kind of important that we, seriously tackle that problem to make sure that half the population is treated, especially at such a critical stage in their life.
Yeah, definitely. I'm just thinking about, you know, like you've obviously studied a lot of things, you've researched loads of stuff, you've heard loads of other research. Is there any, scientific research or things, questions that are unanswered, um, that you're particularly excited about or hope to see, answered in your lifetime?
In the biological world, so one thing that I think is quite exciting at the moment. Um, so have you come across these things called organoids?
Is that in a cell where you have, like, in a normal body you have different organs? Is it cells in the cell you have smaller, like, little organs, and they're called organoids? Is that what they are?
Yeah. So have you seen in the news things like mini brains or brains in the dish?
um I've seen stuff about how they'd get like little brains in a dish to play pong.
Oh, right. Right.
The game where you move up and down and either
Oh,
side and the
that's
ball gets bounced.
yeah. So organoids were this advent from one of the big players was this guy named or is this guy named Hans Clevers
Mm.
in the Netherlands, and he was doing things like intestinal organoids, right? So it's basically you take embryonic stem cells, pluripotent stem cells, and they're able to self-organize. You send in a bunch of inductive cues, you signals, growth factors, and inhibitors of proteins and small molecules that hit different pathways in a combinatorial way, and then all of a sudden they start to organize,
Mm.
and they start to make things that look a whole lot like the anatomy of the intestine. So you have things in the intestine called crypts, which are these little bits where you have the stem cell population sit, and they'll produce like digestive juices and other things to do the activity of the intestines.
Mm.
And then as you come up and out, they'll also make villi, and the villi is where you absorb all your nutrients from. So there's there's a capillary bed that runs up this, and it's one cell layer thick, and everything can diffuse across and go in there, and then that's how we eat, right? So
Yeah.
that's actually how we eat. This is putting food in our mouth. The intestines is how we actually do the thing. Um, and so this was really cool. So we got to study how, things like intestines. There are now lung organoids, brain organoids, um, cardioids, which is heart organoids, right? So they capture features of the anatomy and physiology of organs in the dish. And over the advent of that, developmental biologists are like, okay, cool. That's about the same size as an embryo. we should have some developmental organoids. And
Mm.
so one of the ones that came out, there were a couple, uh, a gastroloid, which is an organoid that looks like they all end in oid, by the way, which is not annoying.
Yeah.
And this represents aspects of the gastrulating embryo. So after you implant into the uterus, you start to, grow and make all these cell types. And that explosion of diversity of cell types is what gastrulation is, right? So this is when we start to make all the different uh, types that will make, your bones and your endoderm, ectoderm, and mesoderm, right? Those are your different tissue layers, right? Inside, outside, and the middle bit. Um, uh, and that was quite cool. So that was developed in Cambridge in a guy named Alfonso Martinez's lab. And now there's a human version, which is being done down in London in Naomi Morris's lab. So she had a big Nature paper not so long ago. Time is a bit of a blur, combination of getting older and the pandemic.
Yeah.
Uh, my hair represents the length of my memory at this point. Uh, and then back in 2021, there's this thing that came out. One of the cool things, so there's a couple different flavors of embryonic stem cells, right? So in 81, the first people to derive embryonic stem cells were in mouse. They took out the inner cell mass, put it in the dish, and they found these things that could make all the different cell types. And so that was Evans, Kaufman, and in the UK, and then what is her name in Texas? Gail Martin. Fun thing, she was the one who named them. Evans and Kaufman wanted to call them EK cells after Evans and Kaufman. They didn't win. I always think that's quite a funny one.
Yeah.
Like, why you don't name the cells after you? Don't do that. But the reason why they did that was because previous to that, there was a pluripotent cell type called an embryonal carcinomal cell, which was pluripotent, but derived from a thing called a teratoma. So these are tumors that have, these are really nasty tumors, germline tumors, where they have like hair and teeth and these other kind of primordial cells that you can extract out. They'll self-renew and give rise to teratomas. So they said, oh, EC cell, well, we're Evans and Kaufman, we'll do EK. That's the story I say, but I don't know what the truth is. So that was done in 81. And then it wasn't until 1998 that we came out with the Jamie Thompson made human pluripotent stem cells, right? Human ES cells. So the mouse flavor represented the pre-implantation version of pluripotency. And when Thompson derived his, he also derived it from human blastocysts, but the cells moved forward in development to a post-implantation version of pluripotency. So it actually had a more restricted version of potential. And so there is a dividing line in development where pluripotency has a different flavor and capacity. And it wasn't until recently that the work of Yasu Takashima, Gagwa, and Austin Smith's group, my old PhD supervisor, they made the human version of the pre-implantation version. And the cool thing about these cells is that not only can they give rise to all of the cell types of the adult organism, but they can also make the extra embryonic tissues, that trophoblast and that hypoblast. Mouse cannot do that. So we showed that in a paper in 2021. That was a paper that I think made sure I have a job now.
Yeah.
Fortunately, thank you, Jenny Nichols and Austin Smith. I appreciate that. And that was difficult and a lot of fun at the same time. In hindsight, it's fun.
Yeah.
But then shortly after that, the work from a group in Australia, Jose Polo's lab, and a friend from Jenny and Austin's group, Ayaka Yanagita, and then after that, Nicola Riveron. These are three papers where they came out with the blastoid, right? So we have the gastroloid, which is that post-implantation embryonic tissue, which is part of an embryo, and then the blastoid, which is the entirety of the embryo. So it looks a whole lot like a blastocyst. And what we can do now is we can start to but we can start to work on a point in development that is completely inaccessible to us, right? And this is implantation. So there are endometrial organoids, right? Tom Rawlings in Cambridge, Jan Bronsons in Warwick, and Emma Lucas, a collaborator here in Sheffield, they've worked on developing endometrial organoids. So we have an organoid that looks like a blastocyst. We have an organoid that looks like the endometrium. We cannot study in utero or in vitro human implantation, but what we could do, and what we're working to do now, is to study this in the dish. That is cool. And this gives us access to a point in development that has been completely untouched.
Yeah. Wow. That's very exciting. And is that, so in your lab at the moment, you said to me before we started about like you set up your new lab. How up and running is things in the lab like?
So I've officially been a member of the University of Sheffield since the 1st of March. So I've had purchasing power for, what is that, 2 months? 2 months? March, April, 3 months. So I'm almost spent all of my startup. Yeah. So I'm just getting a bunch of, I'm hopefully starting tissue culture. I'm doing all my aliquots at the moment. So what you do is you order in a bunch of stuff to make media, and then to make sure that it lasts for a while, you dish it out, put it in the freezer, and then bring it out little by little.
Wow. Okay.
And that's what I'm doing at the moment. So I need to get cells in culture to start doing that. So that's the first thing I'm doing. So I work with this human version of pluripotent stem cells called naive cells that can make those blastoids. And so that's a big prong of work that I'm going to be doing.
That's very exciting. I'll have to come and see your lab when I'm back in Sheffield.
Yeah, absolutely. I was hoping you'd be around because I could come show you like the before and after. What does an empty lab look like, and then what does a functional lab look like?
Yeah. So, you talked about how a lot of the research would be done. I think this is a final question for me now because coming towards the end. But I just want to ask about, a lot of the research would be done on mice and stuff like that. How would that look? Was that something you would do in your lab, or was that, would they just have loads of mice on site and how would they get embryonic cells from mice? Like, how does that work?
Yeah, so it's very well regulated. Caveat, I'm very much pro use of animals in research, if it is for a good reason and you're doing it at kind of the minimum need possible, right? And so there's a massive framework in place to minimize the suffering of animals, while still understanding that one of the best ways to do this work and certain types of work is through the use of animals. And so there are animal facilities where we have trained experts who their entire job is to look after and take care of any model organisms, right? So we to make sure we don't have protesters, I don't think it's a good idea for me to tell you where they are in the university.
Okay.
But we do have animal facilities within campus. Yeah,
that makes sense. Okay, yeah, cool. All right, well, I'll definitely come down and see your lab when I'm back in Sheffield. I should be.
Yeah, absolutely.
September. And I just want to thank you for coming on. It's been very interesting. Have a good rest of your day.
Yeah, excellent. Take care.